So I think we should be starting. I'm really glad that Klaus Nave agreed to give a talk his evening after a full day of working. So Professor Nave is the director of Max Planck Institute of Experimental Medicine in Göttingen, Germany, and has been leading the way in our understanding about myelin and oligodendrocytes. And I am clearly, based also from his talk, we're going to learn something even more new about oligodendrocyte and myelin, and I assume particularly in their role in energy metabolism. Thank you very much, Klaus. So yours.
All right. Thank you so much, Thora, for the kind introduction and also for picking the field of myelin biology for such a neurocentric meeting. So good evening, everyone, from here, from my home office in Berlin, in Germany. It is local time 9 p.m., and that means many people have started streaming videos. So I hope that doesn't affect us as we go along. Now, I have taken from my title that I would like to convince you of a new concept, which is myelin in axonal energy metabolism. And I'm sure you're not familiar with that. So I assume that you're also not working on oligodendrocytes and myelin per se. So let me start very generally and begin by reminding you of what you know about myelin and white matter. And then I would like to introduce you to the concept of glycolytic oligodendrocytes and the role of lactate. I will convince you also that the so-called myelinic nanochannels are very important for the maintenance of our structures, that we can measure the axonal energy balance of myelinated axons in real time. And I would like to discuss something which we think is extremely exciting, namely, is myelin itself an energy store? So let's begin very general and remind you what you all know, that the white matter, specifically in humans, is huge. 50% of the human brain is white matter. You see it in the upper left. White matter in the mouse is also significant. Here it's depicted in black. It's a silver impregnation, but it's much less than that. And these are the long-range connections between neurons and the cortex that communicate with each other over long distance. So these are long axons, also those that go down the spinal cord, as you know. But it's not only the length of the axon, it's also the arborization of axons, which gives it a huge mass compared to the body, and this needs to be maintained. Now, myelin is, as you know, best visualized by electron microscopy. These are very high quality, high pressure frozen optic nerves that we are using to visualize myelin as it's a parallel structure. In fact, white matter, as you know, the optic nerve is a piece of white matter. It's our favorite model system, and you'll see more of white matter biology as exemplified by optic nerve biology, but by extension, it's also true for the rest of the brain and spinal cord. Now, myelin, you also know, is a multi-layered compacted membrane that's made by the oligodendrocyte. It has an ultrastructure that's been visualized best by electron microscopy, as you have seen previously, and it's ultimately made up of lipids and proteins. It's very lipid-rich and has some also very abundant structured proteins. I'll come back to some of them in a minute. It's a very spectacular growth process, if you think about it, that the glial cell stretches to an axon, reaches out, and then physically enwraps its own process around the axon. And it's only actually quite recently with the collaboration with Mika Siemens, we could sort of visualize that this is an actin-dependent growth process of the inner lip that walks around the axon, broadens up, and leaves at the sides the paranodes and the nodes of Ranvier. I would like to stress another aspect, which is not so well known, often forgotten, namely that myelin is not only insulating the axon for electrical purposes and saltatory impulse propagation, but it's also shielding the axon from the contact to the outside world. If you think about it, an axon is 99% deprived of immediate outside contact by the insulating myelin sheath itself. You see this extremely in the peripheral nervous system, where you know it's the Schwann cells that make myelin, but below here you see an image of a single Schwann cell, a so-called PNST fiber preparation, where you may see on the left a node of Ranvier here, and then another node here, and then there is a Schwann cell nucleus at this position. Everything else is completely shielded by myelin. All these notches are so-called Schmidt-Lantermann incisors, but it tells you the axon is shielded and needs to be supported, otherwise it would physically starve. How is that possible? Why are we thinking along these lines? Now, for us, the story began many years ago when we made mouse mutants to understand the role of structural proteins of myelin, and surprisingly, we found mice in which the structural proteins were not there, but the structure of myelin seemed to be there. And then the axon, not the myelin, seemed to develop problems. And these were very characteristic problems. You see this here for the protolipid protein, the PLP knockout mouse, which showed axonal transport defects, which are followed by these swellings. And obviously, these are very unstable structures, and this resulted in progressive axonal degeneration. And this was not specific to this one protein, now also other myelin protein that we subsequently tested genetically. They always showed the very same story, namely a glial and oligodendrocyte-specific genetic defect resulted in secondary axon damage in the apparent normality of the myelin sheath itself. And this was in stark contrast to another mouse mutant that was known for a long time in the field, which made no myelin whatsoever. It's a Chivra mouse that made, I should be careful, non-compacted myelin, but it has basically no compact myelin. And here you see the axons are unmyelinated and they are lacking these characteristic axonal swellings. Now that together means that the oligodendrocyte A in the Chivra mouse is able to support the axon by these contacts, we presume, but overall it means that the oligodendrocytes really have two distinct functions. One is to make myelin, which is the textbook knowledge, and secondly, to also support axons in a curious way, and these two functions, that's important, can be genetically coupled. In many diseases, you have both defects at the same time overlapping. What is the underlying mechanism? Now, a number of years ago, we put forward a hypothetical model based on interesting observations that I will not go into detail about, which suggested that oligodendrocytes that shield the axon are also glycolytic cells, which take up glucose here and then go through glycolysis and deliver lactate through monocarboxylate transporters into the axonal compartment, where mitochondria use that to make ATP. The next slide shows it in similar, again in a schematic fashion. Oligodendrocytes here use the cytosolic space for glycolysis. GLUT1 gets glucose in, and monocarboxylate transporters MCT1 and MCT2 on the axonal side put the lactate into the axonal compartment. This gives you some of the original evidence, which I will not go into. Now the problem is, if there is a massive influx of lactate into this space, then there is a risk of lactic acidosis, because this better be controlled. Otherwise, you would flood in too much lactate into the space. How is that achieved? Now, it turns out that the axonal energy requirement is, as you all know, primarily determined by the spiking activity of the axons. And again, without going into details, we could show that oligodendrocytes themselves express NMDA-type glutamate receptors for the purpose of regulating the energy metabolism. Now, others have shown that a fast spiking axon always sheds or releases small amounts, trace amounts of glutamate, not only the presynapse, also along the axon. And this, we think, is taken up by the NMDA receptor and transduced into a calcium signal, which puts glucose transporter 1, that's the glucose transporter of the oligodendrocyte that I'll come back to again, into the membrane more abundantly, which then results in a higher rate of glycolytic flux, a higher rate of transfer of lactate into the axonal compartment, and more ATP. And in this way, the spiking frequency is sort of miraculously translated into the proper amount of metabolic support that sustains this spiking frequency. Obviously, this can only be part of the story. It's more complex. I'll come back to that as well. But I'll show you here just as an example, how amazing this support system works. If you take cultured oligodendrocytes, you see them on the lower left, and stained for GLUT1, you see the green label is all inside the cell. The moment that you add NMDA, not the moment, but 30 minutes later, everything, all the GLUT1 rushes out to the periphery and helps glucose entry. That's similar to GLUT4 regulation by insulin type stimulation. But here it works for the myelinated, myelinated brain. Now, having said this, the obvious question is, how does lactate reach the axon? I said, after all, it's an insulating problem. And how does the lactate now reach the oligodendrocyte? If you reach the oligodendrocytes, target the axonal compartment. Now, if you conceptually unroll a myelin sheath, you see something what you see on the upper right, namely compact myelin is in the middle, but this compact myelin is, when rolled out like a carpet, will be surrounded by a cytoplasmic tongue, a cytoplasmic space. And this cytoplasmic space is ultimately providing a contact between the connection, I should say, from the oligodendrocyte to the periaxonal space. These would be the so-called paranodal loops here. And this space here, which is cytoplasmic of the oligodendrocyte, is in fact touching the axon itself. So all these spaces where the green arrows point to are cytosolic spaces of the oligodendrocyte that face axons. Now they're in a beautiful position here to then allow the transfer of metabolite from here to there. And these are the monocarboxyl transporters again. And that's what we think is the connection from the oligodendrocyte to the axon. Now I'd like to point out that these proteins also turn over, like all membrane proteins. So it's not only the membrane-embedded MCT proteins here, but it's also cargo proteins, the vesicular cargo that contains MCT proteins that needs to get here in order to turn over, in order not to lose these metabolic connections. On the left, you see here an immunoelectron microscopic imaging of the MCTs as they're sitting here close to the axonal membrane. Now, if that is true, then anything that would disrupt the continuity from the oligodendrocyte to this space here facing the axon would ultimately result in a lack of proper import of these MCT channels and long-term functioning. I'll come back to that. Now unfortunately, we cannot look at this MCT protein as it sits in the vesicle and moves around because we have no proper label, but we have a surrogate for that, which are transgenically labeled peroxisomes. Now we made transgenic mice in which the peroxisomes are labeled by a fluorescent protein that is targeted to the peroxisomal lumen with the targeting sequence. And when we use oligodendrocytes from these transgenic mice and put them into culture, then you see that they make myelin on cultured neurons. And now we can see the cytoplasmic compartment because it's from an oligodendrocyte that additionally expresses a fluorescent marker in the cytosol. But then you see puncta, and these puncta here are single fluorescent peroxisomes inside the cytosolic channel system. We think they represent something like a cargo that could also contain MCTs for the transport of metabolites. Now, I don't have a video, and I'm afraid videos wouldn't transmit tonight, but what I can show you that in a time-lapse series, these cargos really move, and they have a speed of roughly 2 to 3 micrometers per 10 seconds. And this illustrates, I think convincingly, that these spaces are functionally important, and they're also important for moving cargo vesicles around, and by extension, they are important for maintaining the function of the metabolic support. Now, the entire architecture of the cytosolic channel is obviously dependent on the proteins that make it up. And I'd like to point out the CNP protein, which is an antagonist in the cytosolic channels to the myelin basic protein, which helps compacting things. If you don't have it, you see that the compaction becomes extensive, and then you're losing the integrity of these channels. And that's what we think is happening in the CNP knockout mouse, which was the first to show the axonal degeneration phenotype. And what we can show by Western blotting quite nicely is that in the CNP knockout mouse, the Western blot reveals a dramatic loss of MCT1 in purified myelin, which means this is the lack of the continued trafficking of these vesicles. Now, we are not only having a channel closure problem like in the CNP1, we also have a potential channel clogging problem. I would like to discuss with you by looking at another model of a disease which is better known to you, namely the ALS mice, which are caused, the disease that is, for example, genetically caused by SOD aggregation. And you may know this model in which SOD, mutant form of SOD, form aggregates, not only in motor neurons. We can show, they're also showing these aggregates inside oligodendrocytes. What you see here is a cultured oligodendrocyte in monolayer, no axons around, and you're staining not only for CNP, which is the glial protein that marks in green the territory of the cytosolic spaces, which in a cultured oligodendrocytes correspond to the nanochannels, but they also show the misfolded SOD as puncta. When you move in here and look in the enlargement, you see that these puncta are really clogging up this system of of channels. We think this is relevant also for the in vivo situation. Here we're looking by immunoelectron microscopy into the spinal cord and even optic nerve of these mice. We can see very clearly, very nicely, that SOD now is not only in axons as a neuronal protein, it's also in the channels of the associated oligodendrocytes, also here on the right. We think this is relevant because when we look into another line of transgenic mice that Don Cleveland has given us, in which we take out the SOD aggregates from the oligodendrocytes by Cre recombination using SOX10 Cre, which is an oligodendroglial lineage Cre driver line, we can see that the disease phenotype is ameliorated. This is a clinical score which normally goes up relatively steep in this age bracket, but when we remove the SODs from the oligodendrocyte compartment by Cre recombination, you see it's much flatter. I'll also show you, to convince you even more, a survival curve of these mice. And you see here these mice have a career in the natural form. Transgenics have a very predictable cause of death or cause of lethality at around that age, which we can significantly prolong by taking out the SOD expression from oligodendrocytes and OPCs, I should say, using the SOX10 Cre mouse. And what is quite interesting to compare, when we go into the literature and look what the lab had shown previously, using motor neuron specific Cres to remove Cre expression from motor neurons, it's about the same amount of rescue or partial rescue that you can achieve, as we can show here with the SOX10 Cre. What is also interesting, I think, is when we compare this early Cre remover with a late Cre expressing system. This is Morg Cre, which only starts expressing and recombining in mature oligodendrocytes. Then we no longer see this difference. And we think it's best explained because at that late stage, the oligodendrocyte is already filled with mutant proteins, which are going to form aggregates. It's too late. But if we start early in the SOX10 Cre stage, then we're preventing the aggregates to even form prior to myelination. When myelination then happens, it's basically a compartment that is free of these aggregation-prone proteins. So let me switch to another question, that is axonal ATP synthesis really, lactate provision really important practically? And for that, we turned to metabolic sensors, which we have here. And you know how they work. ATP binds at that moment. There's a FRET interaction. We have used these sensors to make transgenic mice in the neuronal compartment, and we're not using the whole brain. We're using optic nerve again, and now can look at axons from the optic nerve. These are myelinated axons taken acutely out of the animal, and we're using them for electrophysiology and imaging at the same time. So when we stimulate them, we can record a compound action potential. There's a characteristic area underneath this compound action potential curve, which gives you as a constant basically the 100% integrity of conductivity of the optic nerve in the presence of glucose or lactate. You're not surprised. That is a very stable situation over time, ex vivo. But when we take oxygen and glucose away, this cap area drops down to nothing within seconds. No ATP generation. You give it back, and oxygen and glucose back, and the cap area goes back to normal. If you prolong, it would do that, but in a short interval it does. And this is very nicely matched with the measurable ATP concentrations in those myelinated optic nerve axons. Now, this is a rather unphysiological experiment. Let's look into a more physiological experiment, which is increasing the conduction frequency or the spiking frequency. And this is, let's say, a physiological range. And when we ramp a stimulation of the optic nerve through this physiological into an unphysiological range, you can see how the cap area drops down, and at the same time also the ATP drops down. Now here we can ask, is this now relevant for lactate consumption or not? And we're doing this by giving inhibitors. We can ask the question, is at very low frequency of spiking, the ability of maintain a cap area preserved in the presence of these inhibitors of MCT1? These are competitive or chemical inhibitors. And you can see at low frequency, there is no inhibition effect. That is remarkable. It requires higher and higher frequencies of stimulation in order to see the effect of these inhibitors. This in reality means that MCT1 blockers matter only if the axon is spiking. So it's the spiking axon, not the resting axon, that is metabolically supported by lactate. And we think this is quite important. Now, that brings up the question again, how is it regulated? Because axons spike or not spike with different frequencies over time. And this system that I'd introduced to you that uses the glutamate signaling is a relatively slow system. It adapts over the course of many minutes, 15, 30 minutes. It's not adapted to prevent excess lactate in the short range. So there must be a way of getting rid of lactate even in the short term. So how does a system like that get rid of excess lactate? If the muscle makes too much lactate, it goes to the bloodstream, you have some muscle ache, but then it's gone. But in white matter, it's not so trivial. Now, we were thinking about this and we found, incidentally, a very remarkable paper, and that was authored by Hugo Bellon, who works, as you may know, on Drosophila. And Drosophila, I should add, has a similar system with glial cells also providing lactate to the axons in the absence of any myelin in Drosophila. And he made the interesting observation that if you have a mutation in Drosophila in which the axonal mitochondria don't work, they don't utilize the lactate or the pyruvate to make ATP, then he found, surprisingly, the glial compartment full of lipid droplets. Now, if we translate this for a moment into this image here into the glial world, it says basically here the axons lack functional mitochondria, and then in the associated glial cells, so this is now Drosophila, there are lipid droplets forming. Now, it's not only in the mutants it's much more. When he looks into the wild type, he also finds them now. You realize that there are some, but there are many more. And this is a remarkable finding. What could that be? Now, it's a way of energy salvage. It's basically detoxification because the pyruvate that is not used is basically decarboxylated. Acetyl-CoA is making fatty acids. The fatty acids are then shuttled back to form these lipid droplets. Now, lipid droplets in a non-myelin organism were an interesting finding. And we wanted to know, what about lipid droplets in those vertebrates that don't make myelin yet? So let's go down in evolution to the fish like lamprey and other fish that do not make myelin yet. Do they also have lipid droplets? We were surprised to find, yes, they have lipid droplets in their fiber, in the axonal fiber bundles, which has no myelin, and they're actually full of big lipid droplets. These are lipid droplets that you see here. Here's reconstructed by serial electron microscopy. And quite interesting, we now think, well, that's an hypothesis. Could myelin now have evolved as an alternative to these lipid droplets? Basically, we're saying, is it that the oligodendrocyte has learned to turn lipids into a smarter energy reserve? Now, this is a somewhat wild hypothesis, and we can never do experiments in evolution convincingly, as you know, but we can ask the question, if this were true, then maybe the myelin, today's myelin, has still some of the properties of the lipid droplets, which are well-known energy reserves. So in order to address that, we made one experiment, which is we took oligodendrocytes and astrocytes, the whole white matter out of the intact brain, using our optic nerve again, and just putting in the culture dish ex vivo and incubated it for several hours. And asked the question now, can oligodendrocytes and axons and, no, not axons, can oligodendrocytes and astrocytes and other glial cells survive in the absence of glucose for an extended period of time? Now, in this particular image, you see the optic nerve, which has been labeled for astrocytes with a GFP label. So these are the astrocytes in the optic nerve. But when we do this experiment and wait for 24 hours, 20% of the cells are dying. You see this by propydium iodide incorporation. These are all the DAPI-positive cells. And guess who is dying? It's the astrocytes. They are all gone. If you look with a similar transgenic label for oligodendrocytes survive this easily. They don't suffer from the lack of glucose. That's interesting. But when you do the co-labeling, see all the oligos that have no glucose are same in number as in the regular medium. But for astrocytes here, you see a big drop. Interestingly, also microglia and OPCs do not suffer very much. So somehow the whole system seems to profit. Now, what is the mechanism behind that. Now, it turns out when we look at a slightly earlier time point, namely 16 hours, this is a time point where all cells survive. So 16 hours is a survival time where oligodendrocytes, no one is dying. That's interesting. So it means the survival mechanism applies to all cells at the same time. When we prevent oxygen from reaching the compartment, then virtually all cells are dying. That means the survival is only possible because something is basically burned, is oxidized. And what is oxidized? Well, when we add to a normoxic condition crotonic acid, when we prevent the fatty acid beta oxidation that way, then you also see the same loss of survival. And that tells us that it's presumably the beta oxidation of fatty acids in the system that at 16 hours not only supports oligodendrocytes, but to some extent also other cells, including the astrocytes, which are ultimately dying, but certainly the oligodendrocytes do. Now, if that is true, that means the myelin is used as an energy source. Can that be visualized? Well, we could visualize this here by looking into electron microscopic images early and late, before with glucose and without glucose. And now the EM quality is no longer great after 16 hours of ex vivo incubation, as you can imagine. But when we score the number of those axons that have very thin myelin, means very high G ratios, then you can see there's a dramatic difference and there's a dramatic myelin loss. That's basically, we're seeing the loss of myelin even after 16 hours already. The real question is this energy usage also good for the axon? Now, obviously, the axon will degenerate when you can cut it out from the optic nerve. But functionally, in the time window that we're looking at, can we see a benefit for the myelinated axon under starvation condition? Now, you already know this experiment, which was taking out the optic nerve, clamping it, stimulating it, and measuring the compound action potentials and recording this over time, this cup area, which you know. Now, we did this now not with 10 millimolar glucose, but we did it with a very low level glucose concentration, namely 2 millimolar glucose. This was empirically determined as that amount of glucose that it requires to maintain at a very low spiking frequency a stable signal. Only when you gradually increase the spiking frequency, meaning the energy requirements, to 1 hertz, to 3 hertz, even 7 hertz, now you see a decline of this. Okay, so this is really a borderline situation of functional integrity. And when we do the same thing now in the presence of the fatty acid beta oxidation inhibitor, which you have introduced earlier, this bromocrotonic acid, now you see that under those limiting conditions, the decline occurs rapidly. Even here, if you were to extend that, that is stability, that is not. So that means under those conditions here, fatty acid beta oxidation participates in the energetic support of axon function as well. Now, this is obviously an ex vivo experiment. By the way, it is also applying to the ATP level, so we can overlay this image with the ATP sensor measurements at the same time, as I've shown you earlier, but you see it's a decline of ATP. But this is ex vivo. Now, the only question is, does it also apply to in vivo? Is it only an ex vivo artifact? Now, we cannot starve mice for obvious reasons. And it's also very difficult to selectively starve oligodendrocytes and not other cells. So we did the next best experiment that we think we could do, and that is we're starving oligodendrocytes in vivo by selectively removing genetically the glucose transporter 1, which you remember was the GLUT1, the transporter that oligodendrocytes require to take in glucose. Now, when we take a mouse that has normally developed and using a tamoxifen-inducible Cree tool, this one, and then take out GLUT1 expression, then it takes a long time for the GLUT1 to also disappear. But we can show this by Western blotting. You see here GLUT1 is after 4 months after tamoxifen administration, it's significantly downregulated, but it takes a long time. GLUT3 on the neuronal side, for example, is unchanged. Other proteins, including MCT1, by the way, is unchanged. These are other markers that are internal markers. But there is a selective difference of GLUT1 expression at that late stage. We don't know for how many days or weeks that is so significant. But we can now ask the question, does that also affect myelin thinning? Does it affect myelin integrity? And here again, you see the results of electron microscopy, and you see, yes, that after that time, we measure the G ratios again, and this is again a measure of myelin thinning, if you like. Myelin is in these conditional knockout mice is significantly thinner than in the comparative wild-type mice. So that is the best that we can do to show that this is of physiological relevance also in vivo. So with that, I think our working model of the dual function of myelin really has changed to and introduced a new player into this. We now look in a way differently at the role of myelin. Myelin is not only there to make the impulse propagation fast in the nervous system, but there is also myelin lipids in oligodendrocytes being a huge energy reserve, which may or may not be utilized, but it's a buffer, an energy buffer, if you like. And compare this for the moment with this little energy that we know exists. This is glycogen granules in the astrocytes. They exist, they are very readily metabolized, as you may know. But overall, the amount of energy reserve in the myelin compartment is much more than in the astroglial compartment. And with that in mind, I personally look at the white matter in the human brain completely differently. I see this as a huge energy reserve. And you may have asked the question, why is the organ that uses most of the body's energy not equipped with more energy reserves that are seen? I think we're looking at them right there, but they haven't been interpreted properly. I mean, this is still a hypothesis. It's an interesting working hypothesis, but I leave you with this idea to think about, and leave you also with a few points that we think are the main points of the talk. Oligodendrocytes provide lactate to spiking axon. This has been seen by others as well in the meantime. I've shown you that the architecture and specifically the continuity of the myelin channels is critical for support. I mentioned the ALS in mice. Some of that is support system is regulated by NMDA-type glutamate signaling. It's not worked out. We don't know how other receptor systems work. There's obviously a function of glutamatergic neurons. There are other myelinated neurons, as you well know. ATP levels in vitro can be monitored. We're trying to go into vivo with this at the same time. I think it's a very nice readout of neuroglia interactions. And finally, I left you with this somewhat speculative idea of myelin itself being a metabolic energy reserve. I have not the time to also go into our most recent area of research where we think, ask the question whether all of this, which I've shown you, is relevant for degenerative diseases of the nervous system, where when the energy balance is low, some of the disease processes, including those that we think lead to Alzheimer's disease, amyloidosis, and things like that, could be negatively infective. And myelin aging by itself could be a very interesting risk factor upstream with these altered energy metabolism that the oligodendrocytes provide. Okay, with that, I would like to stop. I was told we have 30 plus minutes. I'm not going too much overtime. I'd like to thank the team in Göttingen that has been fantastic for many years. I mentioned the unpublished work of Ibrahim Asadullahi, Andrea Trevisiola, and Alexandra Mott. And we have continued collaborations with former lab members, Julia Atker, Johannes Hirling, and Ayman Saab, continued collaboration with others, mouse providers. Very important funding, and I thank you very much for your attention. I'm looking forward to your question. Thank you very much.
Thank you so much, Klaus, for very thought-provoking talk. I just want to remind those who are listed to please put your question in the Q&A. And I'm going to start asking one question. So when you knock out the glucose transporter, and you propose that the oligodendrocytes is almost phagocytosing or eating up the myelin in order to generate, to supply itself with energy. What is the effect or its capabilities to support the axon and the firing rate of the neuron in those conditions?
The mice are relatively normal. I think we're just scratching at the tip of the iceberg. The glial compartment is connected. Oligodendrocytes are connected to astrocytes by gap junction and vice versa. Gap junctions have in this mutant the independent supply of glucose, and they're normally energized. So I think much of the normal activity of the oligodendrocyte is sustained through the astrocytes, the same way as it may go backwards in the early part of the story. So I think we're just scratching at the tip of the iceberg here. And so that explains why there is no obvious behavioral abnormalities. We haven't done fancy behavioral testing yet, but I wouldn't be surprised if there is any that may be at the moment just diagnostic.
Okay, yes, I'm going to continue with a little bit of the question here, and they're popping into in the channel. So here's a question from Maria Ostetrova. Is it known which fatty acids or which lipid species are the most important for the energy storage function of the oligodendrocytes?
Yeah, that's an interesting question. We think it's fatty acids. It's not obviously the cholesterols, which are not degraded in the brain, as you know. They have to be secreted for that. We don't have clear data. We think any long chain fatty acid and very long chain fatty acid is a putative target. We know that the peroxisomes are also part of it. Because the peroxisome conditional lockout mouse, you may have remembered, is part of the battery of mice that we have in which the system fails. And that is also specialized for the very long chain fatty acids. So I think we're looking into regular membrane lipids with the long fatty acid side chains.
Okay, thank you. Here's a question from David Atwell. In what circumstances could the postulated energy reserve in the myelin be transferred to the more energy demanding gray matter?
Yeah, that's a very interesting question. Obviously, how does the energy in the oligodendrocyte compartment reach the axon? Is it ATP? No, it's not ATP. We think there are two ways. We don't have done the final experiment. Either the oligodendrocyte is just more self-sufficient and can just leave more of its regular glycolysis products pass over through the cell without utilizing itself. So its own ATP production would be generated by the pyruvate being delivered to its own, by the acetyl-CoA being generated in its own mitochondria. That's one possibility, and then it leaves more energy for the axon. The alternative, in my view, more likely hypothesis is that the acetyl-CoA is actually built to a new carrier, which is ketone bodies. We think that the oligodendrocytes, which are very close to liver cells in the sense that they make lots of cholesterol, and you may know that the initial stages of cholesterol biosynthesis are related to ketogenesis, can make ketone bodies. And the ketone bodies would be delivered through the very same channel system to reach the axonal compartment. So that's my favorite hypothesis. We're doing the necessary experiments at the moment. They're very time consuming to knock out ketogenesis or keto utilization in the neuronal compartment, but that's what I think is the critical short-range metabolite. We all know that ketones are very well known for long-range communication, but here I think they're a short-range metabolite.
Thank you. I have another question here coming in. In the SOT1 mice, not all axons are equally susceptible to neurodegeneration. Does this imply that energetics of vulnerable axons or their metabolic profile confer vulnerability based on their relationship with oligodendrocytes?
Yeah, I think we haven't even touched this level of analysis yet. I fully agree. We do not understand why we don't know even in ALS to begin with, why does the spinal cord and upper motor neurons that begin with. Obviously, these are energy consuming cells, and those associated oligodendrocytes are part of that. My favorite hypothesis here is that oligodendrocytes are normally supporting axons and they respond to axonal signals, as we have already discussed. At some point of the disease, the oligodendrocyte and maybe also the axon itself fail to communicate with each other. And that is a stage where the system really amplifies, where the problem amplifies, and then the oligodendrocyte is no longer able to listen to the axon and its energetic needs. And that's where the axon goes downhill. That would be my hypothesis. How that is regulated in detail, I have no idea.
Maybe there are more questions to come in. We still have quite a lot of people in the room. I'm a little bit, find it really fascinating, the idea that you're saying that the oligodendrocyte or the myelin is this massive energy reserve. It's a difficult thing to contemplate. So in normal conditions, do you think that when would it be used that the oligodendrocyte would actually start metabolizing the myelin in order to
I, I,
supply
I
energy?
think I think this system would allow, as an evolutionary advantage, to withstand transient phases of hypoglycemia. I think in invertebrate evolution, in mammalian evolution, this is a standard situation in the wild, that you are not properly um, uh, uh, fed. And if the axons if axons degenerate, we know they don't regenerate. So it's critically important that these long axons are preserved from Wallerian degeneration based on starvation. And I think this is a mechanism against that. And I think it's a local mechanism. I don't know what the soma of a neuron has in plan in case of need, but I don't think it has the time to respond to poor situations of energetic support. It's much better to be locally solved as an energy problem. That's where the oligodendrocyte and myelin mass comes in. So I think these are transient issues. We know, for example, in prolonged starvation, for example, in severe cases of bulimia in humans, but also very unfortunate stories of starved people in war situations. There is in the peripheral nervous system neuropathy, well diagnosed, and in the central nervous system we have even MRI detectable loss of white matter. Even CADASIL, which is a disease which you may know based on a mutation, is a local starvation, if you like. And this local starvation is resulting in visible white matter damage. So this is obviously a speculation at this point, but I wouldn't be surprised if this is all fitting together.
That's cool. Okay. But then I have, sorry, because there are no more questions. We have a little bit more time, so I can abuse my power here to question you on the channel. Now we know that some axons have thicker myelin than others. Then is that, so according to your theory, would that then mediate more to their demands of having a reserve of energy?
I haven't thought about it this way. It's an interesting thought. We know that some of our mouse mutants, as they grow old and then show these axonal degenerations, which interestingly takes time, are more susceptible on the small axonal sides than on the bigger, thicker myelinated axon. So I think overall, the energy reserves of the fiber tract is higher if there's robust myelination made. Spinal cord is usually much more robust than cortical myelin. Also, when it comes to the risk factor of diseases like Alzheimer's disease, we know that myelin abnormalities are risk factors. I should have known that I have more time. I could have shared some of the data there. It is always the thin axons that go, and by extension also the corresponding disease markers. Why is it that in Alzheimer's disease the thinly myelinated axons and their neurons are showing the characteristic pathology, never in the spinal cord, where everything is robustly myelinated? So I think there is a myelin connection to neurodegenerative diseases that also relates to robustness of myelination.
Yeah, I think we're kind of at the end. I want to thank you, Klaus, very much for talking to us tonight in the evening. And I want to thank all the attendees for coming to ask the questions and present. We were, I think, up to about close to 50 in the room. So that's good turnout in the evening. Thank you very much. And I hope to see you on other talks.
Thank you all.